Yeah, we are still waiting for the introduction of CAR T-cell in the field of solid cancers. There are many arguments that show that this is not very easy and simple. I mean, there are clinical trials now ongoing that show really promising results, for example, with Clodin, that is currently ongoing. Yet, I think we need to really find ways to translate this to different tumor entities, also using different targets. And I think one of the limitations in the solid tumor field is that we don't actually have so many lineage-specific targets as we do in the hematology cancers. But one of the real lineage-specific targets, in my view, are hormone receptors. So they are expressed on these cells and also on the cancers that derive from these. So, for example, in thyroid cancer, they are expressed on the thyroid cells, of course. But a patient with a thyroid cancer gets rid of the thyroid anyway. So targeting this specific receptor would make a lot of sense, in my view, since you spare all other organs in the body. So this is the reason why we developed a single-chain FV CAR T-cell construct targeting this receptor. And the goal is to develop this further for the treatment of different types of thyroid cancer. We have a very large thyroid cancer here at Halle-University and a very large biobank of tumor specimens. And by analyzing this, we found out that even the less differentiated types of thyroid cancer, not anaplastic thyroid cancer, but poorly differentiated thyroid cancer that is also very hard to treat, still retain the expression of this important TSHR on the cell surface. And so this gives us the opportunity to not only target the relatively easily treatable entities like follicular thyroid cancer or papillary thyroid cancer, but also the less differentiated versions. And since we're a clinical unit, we also would like to develop this really further in the context of a clinical trial once we have assembled all the preclinical models and data.